All right, medical interventions for Thursday and Friday, January 14 and 15. We're going to talk about where you're heading in this Unit 2 work. It's a reminder that at the end of next week, so it will be based on what model that we're in, it would be on the 21st or 22nd, depending on if you're a blue or gold person. If we're still in the hybrid model, will be your test over Unit 1. So make sure you're preparing for that. Yeah, remember, you can use one 8.5 by 11-inch sheet of paper, one side of handwritten notes on that test. So that test will take place, and they'll take most of that class period on that day. So just keep that in mind. But we're going to, remember, we do not have school on Monday, so we come back Tuesday through Friday next week. So where are we at in the course? So you had the 2-on-1, 2-on-2 genetic counseling. And so kind of briefly got started on Tuesday, Wednesday with your genetic counseling patient. And you were supposed to, on your own time, Tuesday, Wednesday, go through this first page. So we kind of just got started. So we will be spending time Thursday and Friday going through your genetic counseling, your patient counseling session with your chromosome board and diagnosing and counseling the patient or the patient's parents, family. So what I want you to focus on outside of class time on Thursday or Friday this week is this last page of the 2-1-1, 2-1-2 assignment, which is copying our genes. And this virtual lab, unfortunately, with Flash no longer working, does not work. So they're working on converting that. So that is not able to do it, but I'm very grateful for this other biomed teacher who has put together a screencast YouTube video of this. And so if you watch this video, basically watch the video, it kind of goes through the steps of polymerase chain reaction that are in here. We'll also talk about it in class on Thursday and Friday a little bit. We'll go through a video in class that is in courses 212. I'm in the wrong lesson. We'll also go through this video, polymerase chain reaction. There's an animation of PCR that's right here. And so essentially, remember, polymerase chain reaction, the whole purpose of it is to produce lots and lots of copies of a specific segment of DNA. All right. And so it's the same process, different cycles that are done over and over and over and over again to produce more. The thing with to produce more of one piece of DNA, right? And so if you're going to do experiments on DNA, if you're going to analyze DNA, it's so small you need lots of it. You need a lot of DNA to be able to, let's say, view it in a gel or to be able to manipulate it or to produce a protein product. And so it also helps us to take one copy, make multiple, so you can do more experiments on it to detect different aspects of it. So taking one piece of DNA and making it endless in how many copies you can make. And so PCR really is 3 stages that are just done over and over again. And so if you look, I'll scan through some of this animation with you guys real quickly. So you have some sort of sequence, and there are the ingredients we'll talk about. You need nucleotides, you need polymerase, and you need primers. Primers are like the starting sequences. And so those are going to hook on. All of that kind of gets mixed into a tube. All right, so you start with your target sequence that you want to copy. All right, and I'll kind of play through this animation real quick. So you basically separate. The first thing you have to do is denature the DNA, which means to separate it. So denaturation. So there's denature, anneal, and extend. So you start with the sequence that you want to copy. You denature it, which means separate it. Then annealing is where you anneal or stick or glue the primers, which are the starting sequences. And so these primers go there, and then you have these nucleotides and polymerase in there, so they will then do extension, and they will add more nucleotides. So now where you had one copy, you have 2. So that's how polymerase chain reaction works. So you start with one DNA sequence, and you now make multiple copies. So now we have 2 copies. And you're going to go through that again and again and again through multiple cycles of it, of denature, then anneal, where you add the anneal to primers, and then extend. So denature, anneal, extend. Denature, anneal, extend. DAE. Denature, anneal, extend. And this just continues, and you get the extension of the nucleotides here any second now. All right, so here you get to the extension. And the polymerase extends with the nucleotides, and you now have 4 copies of that DNA strand, and so on, right? And so this is just going to continue until it can continue to make more and more and more copies. And so and that happens in a thing called a thermocycler, which we'll be using in class. The tricky thing about this is that DNA, these 3 reactions, denaturing, annealing, and extending, happen at 3 very different temperatures. And so we'll use something called a thermal cycler, which is basically you take a really tiny tube and it goes from one temperature to the next very quickly. So that video that I showed you back to here, 211, 212, the 212 video, if you click on this, link, it takes you to a YouTube video. Make sure and you just follow through the steps. The teacher will talk you through this, make sure the audio is on, and you follow through. You'll have a paper copy of this, and you can just answer the questions as you go. And if you have questions, we can definitely go over it. And that's basically it. So really working on this and then just starting to prepare for that test. Make sure you have turned in 1.3 hearing loss. Make sure you turned in the 1.4 vaccination. And as far as assignments, that definitely, like, if you're missing a lot of assignments, make sure you've turned in your superbugs assessment and make sure you've turned in your 116 outbreak analysis, because in the grade book, those 2 are in the assessment category, the same category that your test is going to be in. And so if you have zeros there, that's like just, destroying your grade. So get those in as soon as you can. So you should have turned in the hearing loss and the vaccination, and then the 211, 212 thing is what you're working on. This is not due until Tuesday, 9 p.m. next week. So that is it. Let me know if you have any questions. Reach out to me, and I'd be happy to help you. So thank you.